Hello, and welcome back to Mammalian Anatomy at Cuca College. In this video, we're going to continue our lecture series on chapters 19 and 20, the heart and blood vessels. And here we'll focus on capillaries. So let's talk about them. So what are capillaries? Capillaries are essentially microscopic blood vessels, and they're going to connect arteries and veins. So the heart, arteries, capillaries, and veins actually create a circuit for the circulatory system. And capillaries are going to play a really important role for material and gas exchange between the blood and the interstitial fluid. Interstitial fluid is going to be the fluid that surrounds our tissue. And so it's going to kind of bathe the tissues. And this is where the fluid is going to deliver nutrients and kind of pick up waste products. Very similar to the cerebral spinal fluid that we talked about when we were in our section on the nervous system. So what do capillaries do? Capillaries are going to deliver nutrients, gases, hormones, other components to the tissues that need them. And it's going to pick up waste products, gases, and other hormones and take them to where they need to go and really get rid of those waste products. So let's talk about the structure of capillaries. It is important to note that arteries and veins that we are talking about do not do exchange. Only capillaries allow for this nutrient exchange. So we're going to talk about three different types of capillaries: continuous, fenestrated, and sinusoidal capillaries. These capillaries are going to be 1, found in different areas of the body, 2, be structurally different from each other, and 3, have difference in permeability. Permeability is essentially how leaky the capillary is. The more permeable it is, the more material and fluid and other things can pass through. So more permeable, more leaky. And so let's get into them. So first one is continuous capillaries. Continuous capillaries are going to pretty much be composed of just the endothelium, just the tunica intima, the deepest layer of the vessel wall, and the basement membrane that surrounds it. It's not going to have a tunica media, so it's no smooth muscle, no elastic fibers, and it's not going to have the tunica externa. It's not going to have that outer piece of connective tissue connecting the vessel wall. It's just going to rely on the endothelium and the basement membrane. Continuous membranes are going to, or continuous capillaries are going to be the typical capillaries that are found in our body. And what we'll see here is the endothelial cells are going to be a smooth surface, and there's no holes or any features like that going to be in the endothelium, and that's going to be important when we talk about other types of capillaries. And the cells are going to be tightly close together, like so, here and here, without any gaps or spaces. They'll be held together with different junctions, like tight junctions and things like that, to be able to travel through. Things that are small, uncharged, and lipid soluble will actually cross the endothelial plasma membrane directly via diffusion, or we can actually use endocytosis and exocytosis to allow for bulk movement of material across the cell. But typically, continuous membranes are going to have lower permeability than the other type. Continuous capillaries are going to have low permeability compared to the other types of capillaries that we're going to look at. These capillaries are going to be found in all tissues of the body except epithelium and cartilage. those tissues are avascular, so they're not going to have blood vessels at all. So that's the continuous one. Let's look at the fenestrated. Fenestra comes from French, meaning window. And so these capillaries are going to have windows within their cells. So if we look at the structure over here, we see here that the endothelial cells are going to have pores located within its structure, and more materials are going to be able to go through these fenestrations, go through these pores, and so it will increase the permeability of these capillaries. So more material will be able to go through. So anything that fits will go through. However, it's still important to notice that the endothelial cells are going to be closely attached together along the side. There's not going to be any space or gap between the cells. There will be, however, gaps or fenestrations in the endothelial cells. This is going to be found in areas like endocrine glands, intestines, and kidneys. Think of the kidneys, for example. The kidneys play an important role in filtering our blood, and so these fenestrated capillaries are going to be in our kidneys, so a larger amounts of material are able to be filtered out of our blood and be processed by the kidneys. I always remember my first time teaching. I think it was my first class ever as a professor, and I was teaching about kind of fenestrated capillaries, and I wanted to illustrate a way for students to remember fenestrated capillaries. And the building I was teaching in was older than sin and hotter than hell, because it was such an old building, and even in the middle of winter, the third floor where I was teaching was swelteringly hot. It would probably get up to 90 degrees in the middle of winter because the heating system was so messed up. And so in the middle of winter, we would always have the windows open, and I would give frequent breaks so my students could get drinks of water. So we were talking about fenestrations, and I was trying to explain the increased permeability of these blood vessel walls. And so what I would do is I'm like, if it fits, it goes. So I'm talking about how there's increased permeability and there's these windows, and the windows are open in the building, and I just took the tissue box and tossed it right out the window. And I'm like, so pretend that wall is a fenestrated capillary, and those windows are the fenestrations. If it's small enough, it goes. The tissue box went out the window. Then I think I had like a pencil or something. The pencil or pen went out the window. So I just tossed everything out the window. And students were laughing and having a good time. And then I tried to pick up a chair, and it didn't fit through the window, so it stayed inside. So anything that can go through those fenestrations will go through those fenestrations and aid in the filtration process. The administrator of the building thought I was crazy, and I promptly picked up everything at the end of class, but it was a memorable experience. So yeah. So if you have to remember fenestrated windows, remember the French for a window and tossing things out the window. So it has higher permeability. The last type of capillaries are going to be sinusoidal capillaries. Sinusoidal capillaries will have the fenestrations, just like we saw in the fenestrated capillaries. So you can see these little holes within the endothelium, but the endothelium will not be like continuous. What will happen is that there'll actually be gaps between adjacent cells, and this will allow even bigger things to go through. And the basement membrane here is either very, very thin or absent completely. And so large things that are soluble in water or like cells and things like that will be able to pass through. So where are these things located? One really good example is bone marrow. Remember, bone is responsible for making cells. And so those making cells, specifically blood cells, and those red blood cells and white blood cells actually have to enter circulation. And so they're going to actually enter circulation through these sinusoidal capillaries, going through these gaps between the cells. It's also going to be located in the spleen. The spleen plays a major role in collecting dead and damaged red blood cells. So we're going to need to have these gaps to be large enough to collect those damaged red blood cells and break them down. So those are sinusoidal capillaries. So for these three capillaries, know their relative permeability, be able to describe their structure and compare them to each other, and know one location where the capillaries can be found. Capillaries are not found as just individual vessels. They're actually going to form capillary beds. So what will happen is a single arteriole will come down and it will give rise to many interconnected capillaries that will eventually come into a venule, the smallest veins that exist over here. And so it will create almost a network of capillaries located here. And we need to have this network of capillaries because a lot of the distribution of the nutrients and gases and removal of waste is going to rely on diffusion. And diffusion is relative based on the size of the molecule that is traveling. The larger the molecule, the slower that it will travel via diffusion. Also, it's going to only be able to travel a very small distance. So we're going to need a large network of capillaries to be able to supply the tissues located in the body. At the beginning of these capillary beds, we're going to have precapillary sphincters over here. These precapillary sphincters are going to be circular muscles that can constrict and dilate, and these will limit blood flow to a particular capillary bed, and along with the arterioles can limit the amount of blood flow going to specific areas of the body. So if you ever gotten really scared and your face became pale, what would happen is the arterioles and the precapillary sphincters would actually contract and create that paleness that you may experience as a result of that, because less blood flow is going to the skin as a result. So that is our video on capillaries, pretty short and sweet. Be able to compare and contrast the different types of capillaries. All right, I'll see you in the next video and have a wonderful day. Be good to yourselves. Bye-bye.